Oh, very good evening to all the participants for CPGET 2025 and respected resource person Dr. G.BHAGEERATH Garu, Assistant Professor, Head Department of Botany, GDC Rangasaipet, Warangal District. Please welcome, sir. And I have given sharing option. Please share your screen and start your sessions. Please welcome.
I think screen is visible to everyone.
Screen is visible. If possible, please click on slideshow.
Okay.
Yeah.
Okay. Okay. Shall we start?
Okay, sir. Please proceed.
Okay. In the last class, we discussed about the introduction and importance and different branches of the genetics, and as well as significance. Today, we will discuss about Mendelian genetics completely. So, what is meant by Mendelian genetics? Actually, everybody knows very well the father of genetics is Gregor Johann Mendel. So basically, Mendel is a monk. He was worked as a monk at a priest in Austria. Now it is called as a Czech Republic. He was conducted a series of hybridization experiments on pea plant. That is the experimental tool for hybridization experiments is Physum sativum. That is called as a botany, adavi botany, forest pea plant and pilistuntam manam. So sweet pea antenemo Lathyrus odoratus. Sweet pea is completely different from forest pea plant. So the scientific name is Physum sativum. It belongs to Fabaceae or Papilionaceae. It is a member. And the diploid chromosome number is 2x is equal to 14. So this is also very important. A link, the chromosome number is also linked with Mendel experiments also. And this Mendel first time proposed the basic laws of inheritance. That is a revolutionary change to lay down the genetics branch during the 19th century. And also Mendel was the first biologist to use mathematics to explain his results quantitatively. He never goes to laboratory and he don't know about the gene, chromosome, DNA. But even though he tried to explain about the secrecy of inheritance, how the parental characters are transmitted to offsprings, by conducting series of hybridization experiments for several generations, up to 23rd generations also, he conducted experiments. And one of the beauty in this experiment, he worked with pure lines of peas for 8 years. Nearly 8 years, he continued the pure line hybridization experiments. That's why he was recognized as father of genetics. But when will we know about the inheritance laws? After discovery of Mendelian principles. Again, Mendel principles are rediscovered by three familiar persons. That is Hugo de Vries, Netherlands, Karl Correns, Germany, and Erich von Schermach. These three scientists independently rediscovered the Mendel's work once again in 1900. That is a elucidation of the inheritance laws, and he popularized father of genetics. So this is the background story of Mendelian genetics. And moreover, Mendelian genetics is conferred to classical genetics. He always emphasized on how the parental characters transmitted to offsprings or children's by doing pollination program or fertilization or maybe breeding, plant breeding, doing such type of experiments helpful to reveal the results. And also based on the results, he explained about the heredity, and he explained a character is a dominant one, another character is the recessive. So, and he introduced the concept of factors because he don't know about the genes. The factors now we can call as genes. These each factor govern a particular character. A pair of factors govern a particular character. And this theory, this classical genetics is inspired for the chromosomal theory of inheritance later. So, and also it is a basis, or it is a basis for modern genetic principles also. So this is about the classical genetics and Mendel's contribution. Why he choose pea plant? And mainly he choose the pea plant for his experiments. The Pisum sativa plant life cycle span is very short, and also it is easy to grow and easy to cultivate also. Naturally, the flowers are self-pollinated, but we wantedly do also artificial cross-pollination also. It is feasible in this hybridization experiments. And the offspring number is also very large. Once a sample size of the population is very large, definitely the error percentage is minimized, or error percentage is very, very less whenever we taken a large sample size. That's why whenever he conducted hybridization experiment in the F1 progeny or F2 progeny, more offsprings are obtained. Why? Because one pea pod contains many seeds. That's why the resulting progeny number is many every time. That is a good thing for the advantage in this hybridization experiments. And availability of true breeding varieties also. So these are the main things which is favorable for the Mendel hybridization experiments. You know very well, most of the students knows very well. So many number of characteristics are there in the pea plant, but out of the 23 characteristics, he selected only 7 pairs of the character. Actually, Mendel selected 14 true breeding pea plants varieties. It means Mendel selected only 7 characters in pea plant for carrying out the hybridization experiments. These characters are listed here. You know very well the seed shape, seed color, flower color, pod shape, pod color, flower position, plant height. So that is a symbolic miniatures are there here in this box. Stem length, maybe it is tall or dwarf. A pair of factors which govern a particular trait in the plant. రెండు కారకాలు అనేటివి సంయుక్తంగా ఒక లక్షణాన్ని నిర్ణయిస్తాయి అన్నమాట. A pair of factors govern a particular trait in the plant. So according to the Mendel, a pair of factors contribute a particular trait. That is very important. But later it will be changed. Maybe sometimes 4 factors influence only one character or one trait. So that is a different thing. But according to the Mendel, each character represented by a pair of factors. That's why here genes are units of heredity. According to the Mendelian genetics, genes are nothing but units of heredity. We can explain like that. So in the previous sentences asked about the 7 pairs of contrasting traits. That's why I am said these 7 pairs of contrasting traits are linked with chromosome number. What is the chromosome number? Diploid chromosome number in the pea plant. 2x is equal to 14. 2x is equal to 14. That's why 7 into 2, we are getting 14. That is the linkage, link between the how many pairs of characters are selected, and link with the chromosome number. But he don't know about the Mendel don't know about the traits. Is it clear?
Yes, sir.
Okay. So, and one thing you observe that the responsible factors are located on different chromosomes. That is one of the key factor for the success of the Mendel experiment. మెండల్ తన ప్రయోగాల్లో విజయవంతం కావడానికి ఒక ముఖ్యమైన కారణం ఏంటంటే అతడు ఎన్నుకున్న 7 జతల లక్షణాలకు సంబంధించిన జన్యువులు లేదా కారకాలు అనేటివి ఒక క్రోమోజోమ్ మీద కాకుండా వేరు క్రోమోజోమ్ల మీద ఉండడమే అతని విజయానికి ఒక ముఖ్య కారణం అని చెప్పొచ్చు. ద మెయిన్ ఇంపార్టెంట్ కీ రోల్ ఫ్యాక్టర్ దట్ ఈస్ ద రిప్రజెంటేటివ్ ఆఫ్ ద ఫ్యాక్టర్స్ arranged on different chromosomes. The stem height, the responsible 2 factors are arranged on 4th chromosome. Flower color, the responsible 2 genes, 2 factors present on 1st chromosome. Flower position, 4th chromosome. Pod shape, 4th chromosome. Sorry, it is duplicated. 1 to 7 ravali. 1 to 7. In some figures, it is given as a 3rd, 2nd, like that. So total 7 chromosomes are there. Here repeated 4th and 4th again. It is not like that. It is maybe 2nd and 3rd because 2nd and 3rd number is missing in this list. Is it clear? 4th, 1st, 3rd, 2nd, 5th, 7th, 1st. So in this manner, 1 to 7 numbers are represented here. No repetition of the chromosome number in this table, but it is I think it is mistakenly typed in this table. Okay, this is one thing you can clearly remember. Okay, so during the hybridization process, what procedure is adopted? So generally in the first hybridization procedure, selection of 2 parental plants. Selection of 2 parental plants is first one. So basically the 2 parental plants are homozygous with clear distinguished traits. Then you can before going to the fertilization, we can go for emasculation. This is one of the important program in the hybridization. Emasculation means to prevent the self-pollination, we can cut or removal of the stamens from the diploid flower. So from the bisexual flower, we can remove the stamens. So that's why there is no chance of pollen grain received by the stigma of the same flower. So that's why we can emasculation to prevent the self-pollination and control the cross-pollination between the selected traits. That's why put a bag to cover the carpel region to avoid the unwanted cross-pollination, unwanted pollination. So that's why that is process is known as bagging also. After then, we can select the whatever the pollen. This is called, we can see here, Mendel and pollinated flowers using a paintbrush. We could the stamens to prevent the self-pollination, covered each flower with a cloth bag. That is nothing but is called bagging. So in this way, first we getting first generation, F1 generation or first filial generation. Then F1 progeny self-pollinated, we are getting F2 progeny. In this way, F2, F3 like that. So many number of generations results are recorded and notified by the Mendel. And based on the results, he analyze the results and make it as a ratio, mathematical ratio. This is the complete picture of Mendel hybridization process. Is it clear?
Yes, sir.
Okay. So Mendel, he was conducted 3 type of hybridization process. One is monohybrid, second one is dihybrid, third one is trihybrid also. Monohybrid cross, you know very well, a cross made between true parental plants differ in single trait. A cross made between 2 true parental plants, P plants, which are differ from a single trait. Single trait means, for example, height of the stem we can take as a single trait. One plant grown up to 5 feet, another parental plant grow for half feet to 1 feet. So 5 feet plant we can consider as tall, and half feet to 1 feet grown plant considered as dwarf. So these two contrast characters are governed by pair of genes. Pair of genes nothing but is called allele. Allele contain two genes, sometimes which are dissimilar, sometimes it is similar. Similarity is nothing but homozygous. Dissimilar is nothing but heterozygous. One example, a homozygous tall pea plant crossed with homozygous dwarf pea plant. Dwarf pea plant. So these two true parental plants we can consider as P generation, parental generation. During the meiosis, we are getting gametes. The fertilization between the two opposite sex cells, we obtain zygote. Zygote become as embryo. Embryo later given F1 generation. That is, in the F1 generation, we only one parental character is observed. The one parental character is observed, that is tall. That's why F1 progeny 100% with tall character only. That means in the F1 generation, only one trait, one character is expressed, another one is missing. Which character is expressed? Only tall. So that's why tall symbol is capital T, small capital T. So whenever you see the capital letter, we blindly say that is dominant character according to the Mendel. Okay. Whenever see the small t letter in the allele, that indicates recessive gene, that is recessive characters. So in the F1 generation, all 100% tall plants are expressed. Okay. When F1 plant self-pollination takes place, we obtain F2 generation, that is 3 tall plants phenotypically, 1 dwarf plant obtained. So the ratio is known very well. 3:1 is the phenotypic ratio of the monohybrid cross. So genotypic ratio you know very well. Homozygous tall 1, heterozygous tall 2, homozygous dwarf 1. So that's why 1:2:1 is nothing but genotypic ratio. Among the 7 characters, whenever you choose a character with contrast opposite traits, we obtain only 3:1, 1:2:1. There is no difference. Whatever the character you choose, position of the flower or color of the pod, shape of the pod, whatever the character you choose among 7, we always get 3:1 and 1:2:1. That is the thing happens whenever do the hybridization experiment by the Mendel. So based on this monohybrid cross, Mendel was suggested that law of dominance and law of segregation, two kinds of inheritance laws are explained with the help of monohybrid cross. ఏకసంకరణ ప్రయోగాల ద్వారా మెండల్ 2 అనువంశిక సూత్రాలను గుర్తించాడు. ఒకటి మనకు బహిర్గత సిద్ధాంతం అంటాం, రెండవది అలీనత సూత్రం అంటాం. లా ఆఫ్ సెగ్రిగేషన్ అండ్ లా ఆఫ్ డామినెన్స్. సో దిస్ ఈస్ ద pictorial representation. P parental plants, true breeding, homozygous plants. We are getting F1 generation is heterozygous, all heterozygous tall plants. So in the F2 generation, we are getting what are the genotypes and what are the phenotypes. So 3:1, 1:2:1, you already known very well. So based on this monohybrid experiments, he explained first principle is law of dominance. What is meant by law of dominance? In the F1 generation, one allelic gene masked another. One allele was dominant over the other in the F1 generation. So F1 all are heterozygous. F1 మొక్కలు ఎప్పుడు కూడా విషమ యుగ్మజాలుగానే ఉంటాయి. విషమ యుగ్మజాల్లో ఉన్నటువంటి 2 జన్యువులు, యుగ్మ వికల్పానికి సంబంధించిన 2 జన్యువుల్లో ఒక జన్యువు వేరొక జన్యువు యొక్క లక్షణాన్ని కప్పివేస్తుంది, అణిచివేస్తుంది లేదా నిరోధిస్తుంది అన్నమాట. సో వన్ అలీలిక్ జీన్ మాస్క్డ్ ద అనర్. అంటే ఇప్పుడు క్యాపిటల్ టీ స్మాల్ టీ ఉంది కదా, క్యాపిటల్ టీ స్మాల్ టీ ఉంది అన్నప్పుడు వి వెరీ వెల్ నోస్ క్యాపిటల్ టీ స్మాల్ టీ. ఈ క్యాపిటల్ టీ స్మాల్ టీలో ఈ క్యాపిటల్ టీ జీన్ స్మాల్ టీ యొక్క లక్షణాన్ని ఏం చేసింది? కప్పివేసింది. ఇట్ విల్ బి మాస్క్ ద క్యారెక్టర్. That's why F1 plants only show tall characters. But really small t gene never it loses the ఒరిజినాలిటీ. విషమయుగ్మ స్థితిలో ఉన్నటువంటి స్మాల్ టీ తన క్యారెక్టర్ అది కోల్పోలేదు, కాకపోతే క్యాపిటల్ టీ జన్యువు స్మాల్ టీ జన్యు యొక్క లక్షణాన్ని అణిచివేసింది లేదా బయటికి రాకుండా నిరోధించింది అన్నమాట. సో అందుకే ప్రిన్సిపల్ ఆఫ్ డామినెన్స్ అంటే ఏంటి, F1 మొక్కల్లో ఏ లక్షణం అయితే బహిర్గతం అవుతుందో అదే బహిర్గత లక్షణము, దాన్ని బహిర్గత సిద్ధాంతం అంటారు. ఇస్ ఇట్ క్లియర్?
Sir.
Okay, what is meant by next law of inheritance? Principle of segregation or law of segregation. What is meant by law of segregation? In the F1 plants, when gametes are formed, ఎప్పుడైనా గుర్తుపెట్టుకోండి, gametes సంయోగ బీజాలు ఏర్పడే సమయంలో ఒక అలీల్కి సంబంధించిన జన్యువులు స్వతంత్రంగా విడిపోయి ఒక జన్యువును మాత్రమే ఒక గెమెట్ స్వీకరిస్తుంది అన్నమాట. When gametes are formed, the pairs of allele of heredity factors become separated so that each gamete receive only one kind of gene. అంటే ఇప్పుడు మనం ఇంతకుముందు చెప్పుకున్నాం, క్యాపిటల్ ఆర్ ఉంది, ఎఫ్ ఇది ఒక అలీల్, ఒక క్యాపిటల్ ఆర్ ఒక గ్యామెట్లోకి వెళ్ళిపోతుంది, ఇంకొక క్యాపిటల్ ఆర్ ఇంకొక గ్యామెట్లోకి వెళ్ళిపోతుంది అనమాట. సో దిస్ ఇస్ కాల్డ్ సెపరేషన్. డ్యూరింగ్ ద మియాసిస్ ద పేరెంటల్ క్రోమోజోమ్స్ ఆర్ సెపరేట్. పేరెంటల్ క్రోమోజోమ్స్ అంటే ఏంటి, ఈ క్యాపిటల్ ఆర్ జీన్ అనేది ఏ క్రోమోజోమ్ మీద ఉన్నట్టు, మెటర్నల్ క్రోమోజోమ్ మీద ఉన్నట్టు. ఈ క్యాపిటల్ ఆర్ జీన్ ఎక్కడ ఉన్నట్టు, పెటర్నల్ క్రోమోజోమ్ మీద ఉన్నట్టు. అంటే మాతృపితృ క్రోమోజోములు మియాసిస్ జరిగినప్పుడు ఏమైతే విడిపోతాయి, విడిపోయినప్పుడు మన అలీలిక్ జీన్స్ కూడా ఏమైతాయి, విడిపోతాయి. సో ద ప్రిన్సిపల్ ఆఫ్ సెగ్రిగేషన్ ఈజ్ నథింగ్ బట్ F1 plants at the time of sexual reproduction, the meiosis takes place when gametes are formed, the allelic genes are separated and each allelic gene are received by only వన్ గ్యామెట్. ఒక గ్యామెట్ ఒక జన్యువును మాత్రమే స్వీకరిస్తుంది. అంటే ఒక గ్యామెట్లో రెండు ఉండడానికి ఛాన్స్ లేదు ఎట్టి పరిస్థితుల్లో. క్యాపిటల్ ఆర్ ఉంటే ఇంకో క్యాపిటల్ ఆర్ ఇక్కడ ఉండడానికి లేదు. దెర్ ఈస్ నో స్కోప్. దట్ ఈస్ ఎవ్రీ స్టూడెంట్ షుడ్ రిమెంబర్ దిస్ పాయింట్. ఇంకొక జీన్ ఉండొచ్చు, కాకపోతే క్యాపిటల్ ఆర్ పక్కన క్యాపిటల్ టీ ఉండొచ్చు, కానీ అదే అలీలికి సంబంధించిన జీన్ ఉండడానికి లేదు. సో దట్ ఈస్ ద థింగ్ యూ కెన్ రిమెంబర్ ఫర్ ఎవర్. ఇస్ ఇట్ క్లియర్?
Yes, sir.
Okay, next we can go for next slide. Okay, this is about the Mendel monohybrid experiment. Next we can go for dihybrid cross.